Immunohematology Laboratory class. Today we will be doing the backward or reverse typing. This procedure is commonly done in the blood bank section together with the forward typing. This is used to confirm the blood type of the patient. In this procedure, patient's serum is the sample used, and a freshly prepared known A and known B cell will serve as the reagent. For this activity, you will need the following: marker, 2 empty vials with dropper, blue tips, normal saline solution for red cell washing, 2 EDTA tubes, red top, pasture pipettes, test tubes, whole blood that is type A and type B, and patient's whole blood collected in red top. Also, we will be needing your clinical centrifuge. First, we will subject the patient's blood to centrifugation. This is to separate the serum that will be used in this activity. Tighten the lid securely and set your centrifuge for 5 minutes to effectively separate the serum from the whole blood. After 5 minutes, get the blood from the centrifuge and check for any forms of hemolysis. Also, confirm the name of the patient. Set it aside. Meanwhile, get the 2 whole blood samples labeled type A and type B. These 2 whole blood samples will be used to prepare your known A and known B red cell suspension, which will serve as your reagent for backward typing. Get the clean test tubes and label it properly. RCA, that would be your red cell suspension A, and RCB, that will be your red cell B. Make sure that your tubes are properly labeled. Get the blood type A, using your serological pipette, aspirate 0.5 ml of the whole blood and transfer it directly to the clean test tube labeled RCA. Get your blood type B, again using your serological pipette, aspirate 0.5 ml of the whole blood and transfer directly to the test tube labeled RCB. Now, we will proceed to red cell washing. To do this, get your NSS, fill the test tubes up to 3/4 level. Now, subject this to centrifugation. Set your centrifuge to 5 minutes for the first washing. After 5 minutes, get your red cell suspension and decant. Now we will proceed to the second washing. Get your NSS and fill the test tubes up to the 3/4 level. Subject it to centrifugation. Again, Set your centrifuge to 5 minutes. After 5 minutes, get your red cell suspension. As you can see, the NSS is clearer. Now decant the NSS. And then we will proceed to the third washing. Get your NSS, fill the test tubes up to 3/4 level, and then subject it to centrifugation. Set the centrifuge for 5 minutes. After 5 minutes, get your red cell suspension. Again, decant the NSS. And we will proceed to the fourth washing. Fill the test tubes with NSS up to the 3/4 level. As you can see, it has a similar color to a tomato red. Subject it to centrifugation again. Set your centrifuge to 5 minutes. After 5 minutes, get the red cell suspension and decant. Usually, we do a known red cell A and known red cell B by adding NSS to reach a tomato red color. Set aside the washed red cells. Meanwhile, get the 2 empty vials and label it correctly. Label one vial as known A or red cell A and the other one as known B or red cell B. Next, get the tube of washed red cell labeled A and fill with NSS up to the 3/4 mark or until a tomato red color is reached. Mix well. Transfer the mixture to the vial labeled red cell A. It is important to note the date and time of preparation. Same with red cell B. Get the tube of washed red cell labeled B and with NSS, let it reach to 3/4 level or until a tomato red color is reached. Mix well. Transfer to the vial labeled red cell B. Note the date and the time of preparation. Now, you have your prepared known A and known B to be used in the reverse or backward typing. Now, we will proceed with the reverse typing proper. Get 2 clean test tubes and label it with A and the other tube with B. Set it aside. Get the patient's blood sample. Please make sure of the name of the patient you are typing. Aspirate the serum using your pasture pipette. And then transfer 2 drops of serum to test tube labeled A and test tube labeled B. Now you have 2 drops of serum in each of the test tubes. Get the known A red cell suspension and add 1 drop to the test tube labeled A. Mix lightly, and then get the tube labeled B. Add 1 drop of known B cell to the said test tube. Take note of the number of drops you are adding to each of the mixture, as it will affect the degree or the reaction of agglutination. Set the speed of your centrifuge to 1000 and the timer to 1 minute. Place the 2 test tubes inside the centrifuge. After which, you have to get the test tubes and mix it lightly. After which, you have to check it under the microscope. Please take note that you cannot transfer the mixture onto the slide and check for possible agglutination. Macroscopically, you can see that there is no evident agglutination. For the prepared known A and known B cell, these may be used for 8 hours. However, you must keep it inside a refrigerator.